Hey everyone, welcome back. So now we're going to talk about how we control gene expression. So gene expression again is a production of proteins, and there's two steps, transcription and translation. So all cells have to be able to control their own gene expression. So think about a single-celled organism. One cell has to be able to express different genes at different times. A multicellular organism like a human has multiple cell types, and they each have their own functions. So each cell type differs in both structure and then in function. And actually in anatomy and physiology, that's a driving thing that we always push on our students. We drive home this idea that structure determines function. And this is red, apologies, but that's okay. Structure determines function. So each cell has its own protein combination that will separate it from all other cell types, each cell type, I should say. So we have cells that are muscle cells. We have cells that are nerve cells. We have cells that are epithelial cells. We have cells that do the job of making mucus. We have other cells that do the job of diffusion. So different cells do different jobs. And so within those cells, they have to be able to produce certain types of proteins that are specialized to that cell's functions. Now, in general, all cells, or in general, most cells, have genes called housekeeping genes. So these are genes that allow the cells to do basic functions. So in general, housekeeping functions would be things like producing ATP. So in general, all cells have to be able to have ATP because ATP is the cell's energy currency. Metabolize carbohydrates. So they're going to have to be able to metabolize carbohydrates so that they can produce the ATP. You're going to have cells that function in reproduction. So they're going to be able to go through mitosis very readily. That's like your epithelial cells. They have to be able to go through mitosis because we're constantly producing new skin. Think of stuff like that, okay? So again, housekeeping genes are going to be turned on all the time, whereas other genes are going to be specific to the cell type. So here we have red blood cells. Red blood cells have their housekeeping genes on, but they also have production of hemoglobin on. So red blood cells need hemoglobin. Muscle cells don't use hemoglobin. Pancreatic cells don't use hemoglobin, so they're not going to be turned on. They all have a full copy of all the genetic information, but only certain genes are turned on at certain times, okay? Muscle cells have myosin. Pancreatic cells can produce insulin. So they all have their own specializations within the organism. So let's look at gene expression in prokaryotic organisms. It's really easy to understand in prokaryotes. Prokaryotes can use different types of carbohydrates for energy. And so we'll look at E. coli, Escherichia coli. You've probably heard of him as E. coli. I am going to change the color because I like to use a darker color just normally. So E. coli uses multiple sugars as their carbon source. So they tend to prefer glucose. Glucose is blood sugar, so it's the sugar that's in our own blood. And glucose is really easy to break down, so it's the favorite food for E. coli. But they can use other sugars or carbohydrates as well. Specifically, they like lactose. So if there's no glucose but there's lactose, they're going to use lactose. So they can adjust gene expression based on which carbohydrate or which sugar is available. So if they have lactose and there's no glucose, then they can turn on the lactose genes to break down lactose. And this is actually in a functional group called an operon. So an operon is a cluster of genes that codes for proteins related to specific pathways. And so operons have a region known as a promoter region. They have then the operator region, and then they have structural genes. So the promoter region is where RNA polymerase binds. So I'm just going to write RNA poly, okay? This is where your RNA polymerase is going to bind. The operator site is, let me just do this, promoter. And this is operator, okay? So the operator site is where you have, I'm just going to do it this way. I know this isn't the shape of one, but where you have a stop and go light, basically. This is where a protein will either bind or will not be bound. And that will determine if this gene over here, the sequence of proteins over here, is going to be able to be produced. So again, you have the promoter sequence is here. You can see it right there. This is where RNA polymerase will bind. The operator sequence is here, and this is a repressor protein. So if the operon is going to run, this repressor protein has to leave. So when the repressor protein is bound, it stops this operon from running. But if lactose is available, then lactose actually binds to the repressor protein. So it's there, which means, hey, we have lactose in the environment. We can make the enzymes to break lactose down. So when lactose is available, lactose binds to that repressor protein, and the repressor protein moves off of the operator site. And then the RNA polymerase can run along and produce the functional proteins to break down lactose. So eukaryotes also have mechanisms for controlling gene expression. So in prokaryotic organisms, a single promoter can be used to transcribe multiple proteins. In eukaryotes, a single promoter functions for a single gene. And so we have lots of different regulation mechanisms to control the function of our genes or the production of our genes. So here is the list of mechanisms. I'm going to go through this list, and then I'm actually going to stop this video and we'll go into each of these in our next video. So we have pretranscriptional control. So this is prior to transcription even occurring, we're going to stop it or we'll start it. Transcriptional control, so during the transcription. Posttranscriptional control, translational control, and posttranslational control. Okay, so we'll talk about each of these in our next video. Bye.